The broadcast is now starting. All attendees are in listen-only mode.
Welcome to PEI's Masterclass Climate Change in the Bottom Ocean Community. We're going to wait just a couple of minutes for a few more of our attendees to sign in, and then we will get started. So just stay tuned, and we'll be with you in just a couple of moments. Okay, welcome to PEI's Masterclass Climate Change in the Bottom Ocean Community. Thank you for joining us to learn more about polar science and education. My name is Betty Trummel, and I will be the moderator for tonight's class. This course will focus on recognizing the benthic community and understanding how climate change affects its structure. The benthic community includes organisms, both flora and fauna, that can be found either attached to sediment environments or slightly moving around the bottom of the ocean. We must understand how food webs work and how food sources can be modifying due to climate change and what their effects are on the ocean ecosystem. The Masterclass series is designed with two audiences in mind: educators who want to build their science knowledge and researchers who are interested in improving their science communication skills. This class will provide a deeper understanding of ways that scientists and educators can work together. Our next class will be offered in the first quarter of 2019. Details will be announced in early 2019. You can learn more about Polar Educators International on our website. If you have not yet joined PEI and are taking part in this masterclass, please join us. Help us grow our worldwide network of scientists and educators. As you can see, our vision statement: Polar Educators International is a vital international network of educators and researchers aiming to provide a deeper understanding of current polar science. PEI represents trusted leaders working to inspire appreciation and knowledge of the polar regions, their connectedness to all of Earth's systems, and importance to all humans across latitudes and cultures. One of the ways PEI is working to meet the goals of our vision statement is to sponsor master classes like this one, focused on the needs of a dual audience, educators and researchers. We are excited to present the class tonight, and as I said, we'll have another one scheduled for the first quarter of 2019. In terms of questions for the presenters this evening or this morning, wherever you may be, please write your questions in the questions box. We will pose some of them at the end of each of the presentations. Your questions will only come to me as the moderator, and our assistants tonight will try to fit in as many as time allows. Any unanswered questions from the webinar or new questions can be posed and answered in the discussion group in our Google community from the 28th of September, tomorrow, until the 12th of October, which is a 2-week time period. It's my great pleasure to introduce our first speaker today, Dr. Francine Elias-Piera. Dr. Elias-Piera has worked with biodiversity, distribution, and trophic ecology of Antarctic benthic communities since 2000. She is currently a postdoctoral researcher at Korea Polar Institute, leading the benthic food web research line in the project JAMP 2050. Francine has been involved with education and outreach since 2010 in the Council of the Association of Polar Early Career Scientists, APEX, Brazil, Vice Secretary of APEX in Spain from 2012 to 2014, co-chair of Education and Outreach Committee of APEX International from 2013 to 2015, and she is currently collaborating researcher in education projects from a university in Brazil. Our second speaker will be our educator, Dr. Eileen Schwambach. Dr. Schwambach developed her PhD research on science education, chemistry of life and health at the University, I'm going to pronounce this wrong, Federal de Rio Grande do Sul, and did her sandwich PhD in UCL in England. Currently, she works as a high school teacher and also works as a mentor of several environmental education projects for teachers. Eileen is a counselor of Apex, Brazil. She was a Brazilian delegate from the United Nations Framework Convention on Climate Change at the Conference of Parties in Paris, France. The activity that Dr. Schwambach introduces in this webinar will be available in the members area of the PEI website during the online discussion class. It will also be shared on the PEI group Facebook page and then in the discussion group following the webinar. Just a few reminders about the masterclass. Join online in our discussion as part of our Google community. The discussion forum will take place for the next 2 weeks through October 12th. To earn a PEI certificate of participation, either take part in the live webinar happening now or the one that's archived, which will be posted soon on our PEI website. Introduce yourself in the discussion forum in our Google community and take part in discussions regularly over the next 2-week period. See the detailed syllabus on the website and please complete the pre and post surveys. So I'd like to turn this over. I'm going to switch screens and let Francine start with her presentation.
Hello, good morning. Oh, good afternoon.
Do we see your screen, Francine?
Yeah. Okay. Can I start?
I don't see your screen. I want to be sure that we have.
I see it. It's up there.
Do you see it? It's up there? Okay, great.
Okay.
Oh, there we go. Okay.
Hello, everybody. My name is Francine. Hello, Betty. Hello, Betty. Thank you for inviting me. And today we will talk about benthos, the forbidden in the world of melted ice. Well, to start, we have to explain how do we divide the organisms in the sea. We have three divisions, like the plankton, the organisms that they goes with the currents. They don't have many movements. And the nekton, normally the big organisms like turtles, whales, fish that can swim actively in a body of water. And after we have the benthos. Benthos is the community of organisms that lives on, in, or near the seabed. On, in a substratum, sorry. Where can we find benthic communities? We can find in the river, on the bottom of the river, and on the sea we can find in the intertidal zone, mangrove, the coral reef, the deep sea, the ship hull. Here in the deep sea, for example, we have many polychaetes that we will see what is polychaetes. And we can find benthos in other organisms, like whale. We can find here a crustacean, the barnacle, or we can find on the sand, or in the sponge, or in algae, organisms, little organisms in the other organisms. All of these are organisms, benthic organisms. Sorry. Then, below the ice in Antarctica, there are more than penguins and seals. We have a very rich invertebrate community. We have different types and different colors of benthic organisms. Here we can see sometimes the organisms near, like this, near the seabed, we can call benthic organisms too. Then I will show you many organisms that take part of benthos, and you can see the diversity and the rich community. Most of the pictures I took in my expeditions, but we can find other organisms in Antarctica. Okay, I just put main of my pictures here. Then we start with the macroalgae. It's an organism that stay on the seabed or on the rock. The sponges, we have many of them, different types of sponges. We have the pantopod called sea spider because it's a Chelicerata. It's the same subphylum of spider, the common spiders. And we can see many worms, different worms. This Nemertea is an important worm because it's a top predator in Antarctica. And we have, polecates. These. We have depositivorous, we have suspension feeders, and we have carnivorous polecates. We can see many crustacea, like the amphipods, isopod, and comation in Antarctica. Many of amphipod, many species like fish or penguins can eat amphipod. We have many mollusks, different types and colors and size. We can see. Echinoderms, the crinoidea, the sea urchins, the holoturians, the sea cucumber. No, we have different of them. Here the pig sea cucumber, and Ophiuroidea, brittle stars. This is one that we just find in Antarctica, and we have the sea star. many of them carnivorous. This is a top predator too, very important. And here we can see the slow movement of sea star. They can move with the ambulacrarium feet. And here on the movie, we can see that they can respond of some disturb on the water. You know, they have muscles because ascidians are an evolutionary link between invertebrates and vertebrates. And we can find some different fish and other organisms that we don't know what is, not yet, not identified. And to sampling the benthos, we need some sampling machine like the dredge. We can see here come with many sediments, and then we have to wash and literally find the organism on the sand, on the sediment. And we can collect the samples with scuba dive. And here we can see the net and many bottles to bring the organisms, the sediment, to study this. Then we have the box cutter. Yes, it's a box, a literal box. We can put on the water and read on the bottom, and then bring the sediment for us. Here we can see some organisms on the bed, on the sediment. Okay. The benthos, they take part in the trophic relationship, the food web. And remember what is the food web, food chain. We have the primary producers in the terrestrial food chains. We have plants, and in the sea, well, we have the phytoplankton, the macroalgae, and then we have the primary consumers, the herbivores, and after this we have the carnivores, secondary consumers, tertiary, quaternary. And in Antarctica, many times we just see a simplified trophic chain, just with plankton and nekton. But benthos is always forgotten, but it is important in the remineralization and important in the carbon sink, because benthos is the link between plankton and nekton. Many fish and birds and other invertebrates can eat benthic organisms. Then they can make this link between this part, the pelagic part, and the benthic part. we call this benthic pelagic coupling. And then understand how is the environment, the Antarctic environment. We have here in Antarctica the permanent ice cover and the marginal ice. This marginal ice, it forms in the winter and deglaciate in summer. No, and then when the ice melts, the microalgae, the phytoplankton, starts a bloom, starts growing, and then we have many microalgae, and we start with many zooplankton growing here, and this reach the bottom, reach the benthos, and benthos eat this phytoplankton. And here we don't have many phytoplankton. Okay. Then we call these particles like phytoplankton, detritus, like particulate organic matter. Then the detritus, like phytoplankton and zooplankton, or even fecal pellets and bacteria associated to the sediment or to other organic matter like detritus. Okay, the phytoplankton, we have some zooplankton. They can make vertical migration, and then they can go from the up until the bottom, and in this vertical migration the benthos can eat this zooplankton. And here we can see the POM on the water. And to study all of these, the food chain, the POM, we use the trophic markers. They are organic molecules that do not suffer significant alterations in their original basic structure when they go from one trophic level to other. What is trophic level? Is the primary producers, and after primary producers is a one trophic level. Then we have the primary consumers, another trophic level, and second consumers, another trophic level. And then these chemical, these organic molecules don't change on the trophic levels. Then normally the studies, the previous studies that we have the direct tools, there are studies of the stomach, the gut content of the organisms. But imagine I can make the acidian regurgitate. It's very difficult. Then to study difficult areas and difficult animals, we use the indirect tools, the stable isotopes, the fatty acids, and biochemical balance. But what is stable isotope? We have here two atoms, and remember that the atom is made by the negative and positive charge, the electrons, proton, and the neutron that is without charge. No? And then on the carbon we have 6 negative and 6 positive and 6 neutrons. Okay, but we have a similar carbon with one more neutron. This neutron doesn't change the element. The carbon 12 and carbon 13 is the same element, but just change the weight. One is we call light and other we call heavy. Okay, they are not radioactive. They are stable because this is the name. Okay, and then the more abundant, the abundance of each stable isotope. Here we have many of the oxygen 16, nitrogen 14. Then we can see that the more abundant are the light isotope. Okay, then the light here it's more abundant than the heavy. Well, but sometimes one neutron makes many difference. Okay, the heavy stable isotope many times it's very slow to reach and to come to the organism. For example, the plants make the photosynthesis, but the CO2 enter on the plant, okay? But there are more light isotope, and here just one has isotope. Then the body normally have incorporate light isotopes, okay? And then the plant make the photosynthesis normally, and the organisms come to eat the plant. Then the first ones to go to the organism, to the animal, is the light isotope. Okay. Then to the carbon analysis, we use this formula, and we describe the carbon in parts per million. And the isotope on the plant, for example, the macroalgae, reflect the characteristic of the environment. And the isotope composition of the consumers reflect the composition of the primary producers. Okay, and we have differences between the carbon value in freshwater plants and phytoplankton and macroalgae. And we have difference between the phytoplankton in pelagic, the overshore, and in littoral, in near shore environment. Then we can see the same, the phytoplankton, a primary consumer and a fish, and here the same, but with different values of carbon. And then to nitrogen, we use the same thing. We can see that for the primary producers to primary consumers, we have we accumulate the nitrogen, for example. Okay, we can see here on the terrestrial and the water, the seawater. Then we use the stable isotope to meet the main food sources and to identify the position of the organism in the food chain. Then here we can see that the food source for this zooplankton is this phytoplankton. Okay, and we can see the trough position. And the advantages, like I told, it's very difficult to make acidians regurgitate, and many times that we open gut content, we cannot identify the organic matter. Here we can just identify the macroalgae by the cell wall, but here we have detrital matter not identified. And the isotopes reflect the real food assimilation, not the ingested food. The other trophic marker is the lipids, and the lipids are energy-rich molecules. They are used for energy storage. And we have the fatty acids that are the most important component of lipids. The fatty acids are made by long chain with carbon and hydrogen. There are fatty acids with just 4 carbons, and we have fatty acids with 24 carbons. And all the fatty acids has this carbon group, carboxyl group, okay? Carbon, oxygen, and hydrogen. We have different types, the saturate, the monosaturate with this double link, and the polysaturate with more than 2 double link. We have the essential fatty acids. Normally this it's not synthesized by the animals in the body. They have to obtain by food, from food. Normally the essential fatty acids come from plants, but we have a fish here. Why? Because fish has many fatty acids that we can call omega 3 and 6, the more common. And we can find on the fish because the accumulation. When the fish eat the algae, they accumulate this type of fatty acid. Okay. And we have some fatty acids that modify their structure on the body. Then we can eat some food with many omega 3 and 6, but on the body, they will change for other fatty acids. For example, the omega-6 here, they change for here, and the omega-3 here will modify until here. The carbon C24 and carbon 25, they take part of an anti-inflammatory molecule. Because of this, the essential fatty acid is very essential because this is the name. Okay. And the fatty acid composition of organisms varies widely with species, habitat, light, and salinity. Because of this, we can use the fatty acid to identify the food source. Here we can see the diatom normally have more abundant the C16 and C25. What is this? Is that diatom has many fatty acids with 16 carbons and many fatty acids with 20 carbons. Okay, the dinoflagellates has C18, C18-1, C22-6, and the macroalgae 16, 18-1, 24, 25, 22-6. And here I put the copepod. It's an animal, a crustacean, and we have carbon and C20 and C22-1 specific to copepod, okay? Because they eat, and in the body they change and form these two principal, these main fatty acids. And this is the tracer, the signature of copepod. Here we can see macroalgae, the fatty acids of macroalgae. And here we can see the fatty acids of amphipod that eat macroalgae. Okay. Then the C16, C18-1, the C25, C24, C22-6, and here we can find the same fatty acids on amphipod. This can show us that amphipod eat macroalgae. They can eat another things, okay, but the main food is the macroalgae, okay? Then the main good tool for the interpretation of trough position is fat acids, but combined with stable isotopes. When we use the fatty acid, just the fatty acid, okay, it's good to understand. But if we combine with stable isotope, we can interpret the results better. Okay. And another thing, the fatty acids, the composition can change with the habitat, the light, the salinity, and can change with the environmental conditions. Because of this, we can use the two combined to understand the food chain, the food web. And the biochemical balance and fatty acids, mainly the lipids, we use to understand how the different organisms invest their energy, how they adapt to the environment. For example, the same amphipod that eat macroalgae, we can see the energy storage here in February, March, April, May, June, July, and October. We can see a accumulation of fatty acids here until winter. And then when they don't have many food, they use the energy until the summer. And then in the summer, the energy probably will grow again. Okay, then the loss of sea ice affects the benthic community. Yes, the most important change was the decline and the great collapse of the ice shelves. It's the permanent ice, and we can see the oceans are relatively warm, between 1 and 2 degrees more than another years ago. We have a special place called Larsen Ice Shelf. The Larsen is this place on the peninsula. We have the Antarctica here, and here is Larsen. We have Larsen A that collapsed in 1995. Larsen B collapsed in 2002. And now Larsen C, we can see the new giant iceberg. The last year, the Larsen C start to collapse. Okay. Then, with the ice shelf retraction, without this ice, we can expose areas. We can study new areas of the sea floor and change the ice shelf retraction can change the physical and chemical properties of the water column. And this influences the process like sedimentation, fauna distribution, food availability, presenting opportunities for new benthic colonizers to become established. Remember the environment, the Antarctic environment. We have the permanent ice and the marginal ice. Here we have many phytoplankton, and here we don't have many phytoplankton. Then Larsen was this situation, and without ice, with the ice melt, the collapse of the ice, we have this situation now. Then the bent community on Larsen will change in the future years. Here I put an example with a study between with organisms from King George Island, an open place without a permanent ice, and here organisms from Larsen A, Larsen B, and I start from Larsen C. Okay, we can see the stable isotopes that now you can know what is. Here we can see the sediment. Okay, the sediment, it's like phytoplankton here, and we have the organisms here. We can see here link with sediment and here link with sediment. The other organism, it's very far from sediment, and then we can see that they don't eat sediment or the particles in sediment. And see the same species in different places. We can see the difference between the stable isotopes. We can see the difference between the food source and the trophic position. Here, King George Island, Larsen A, Larsen B, and Larsen C. The same with the other species. And here we can see just the nitrogen, the trophic position. Here we have the organism near the sediment. Maybe these sediments are rich in phytoplankton, and in Larsen A, B, and C, not rich in phytoplankton, but in detritus. Because of this, the organisms are more carnivorous than in King George Island. And we can see the energy storage in the same species. In King George Island, Larsen A, Larsen B, and Larsen C. We can see that there is less energy storage in Larsen B and C. Two hypotheses. One, that the food quality is not good on Larsen B and C, and because the organism has to use the energy to find food on Larsen C and B. Then, if we don't have the permanent ice, the environment will change, and then we have organisms change the food, the food chain, okay? And other things that can affect the benthic community is the ice scour. The ice scour is the iceberg. They crush on the sediment and can devastate the benthic community. Because the sponges and other organisms here, they cannot move and have a low metabolism. Many of them, like sponges, take almost 100 years to grow 10 centimeters. Because of this, the ice scour can devastate the benthic community. And we have the alien invasion. With the warm water, the 1-2 degrees warm, the currents from warm place can reach Antarctica and can bring organisms, new species. Species that live in cold water, but not so cold water, not a cold like Antarctic water, but with the 1-2 degrees warmer, these species can reach Antarctica. and we have some crabs, predators crabs, that can eat sea stars, mollusks, and sea cucumbers. And the organisms in Antarctica, they don't adapt to this type of predators. Okay, and this is the chance on the benthic communities that can occur. Okay, thank you. I hope that you understand about the benthic community.
Okay, do we have any questions for Francine before we move on to our educator presentation?
I had a quick question for her. This is Betsy. When you showed those pictures way at the beginning of underneath the sea ice, some of the organisms that were actually in the water, not in the lab, were those taken by a diver or were they taken with an underwater robot?
Sorry, repeat, please.
So towards the beginning of your presentation, you showed some of the life that was actually in the ocean. They weren't images of life that was in the lab. And it was underneath the sea ice. Were they taken by a diver or was it an underwater robot or a camera, a remote camera?
And with a robot. Yeah.
Cool.
Yeah. With a robot, because the images are from 200 meters, more or less, around the. Okay.
Great. Thanks.
Welcome.
Any other questions? Okay, then I'm going to change the screen so that Eileen can begin her presentation. And thank you, Francine. Thank you very much.
Hello, everybody. Can I start?
Yes, absolutely. Go ahead and start. Thank you.
Okay, so thank you for your invitation. So my name is Eileen Schwambach, and let me introduce myself and my colleagues and friends. So I am from the south of Brazil, and here I am a science teacher. I work in Instituto Ivoti and also in Apex, Brazil. And Francine was talking to us from Korea. And we also have Silvia Dota that is talking with us from the southwest of Brazil, from São Paulo. And Francine and I, we did 2 years ago the translation from this book called Celebrating Antarctica, organized by Julie Beckman and Alan Pope. And since that, we start talking and working together in Apex and in this kind of project. So today, what I am going to talk with you is the way we can take Francine's studies, Francine's field, and how we can get this idea and take this idea to the school and show to the students how they can learn more about benthos and about Antarctica. So the first thing I introduced for you, it's a brainstorm. Probably you already know something about brainstorm. It's a very interesting idea to know what the students know about what we want to learn. So we can do a brainstorm using 3 main words. First of all, I think it's interesting to use Antarctica, second, benthos, and for the third, plankton. So we can just put in our board these 3 words and ask for the students, what do they know about Antarctica, benthos, and plankton? And they can make some draw, they can make some notes, They can connect some ideas, they can talk in small groups and make some notes on their notebooks, for instance. And then, so for the first question, where is Antarctica? It's interesting, we always need to show this global map and try to show for the students where is exactly Antarctica, and after that article, for instance. After that, this kind of question like, what is plankton and what is benthos? And also, where did you heard about it? So what is plankton and benthos? It's really interesting, the kind of answers I used to have here from my students. I developed this class last year, and this year I also did the same question about plankton. And look what I always have as answer. I always have SpongeBob. So usually they know what is plankton because of this cartoon. So they always make some connection about SpongeBob, SquarePants, and then they know that plankton, maybe he had an eye, maybe he talks something, and maybe he discussed some ideas with SpongeBob. And then we have a huge discussion about what is really a plankton. So I use the same picture as Francine showed for you, showing that plankton we have here on the river, on the sea, and then this plankton, then we make a discussion about also what is nekton and then benthos here. And I show for you some ways to engage class discussion, like discuss the ideas on the board. It's really interesting. Also showing real pictures after the cartoon. So if we show SpongeBob, it's very interesting to show real pictures after that. Also show what plankton looks like under the microscope. So if we take a sample of water from a river you have in your city, from a rivulet, and then you take this sample of water, you can wait for 2 days, for a week, and then they start reproducing. And then you have just one drop of water with so many microorganisms that you can see with your students. And after that, you can do a concept mapping, showing how students make some concepts and how they connect these ideas. Here I asked for Francine some of her nice pictures, and here we can see, for instance, benthos. We can see the different kind of sizes, the different kind of colors we have in benthos community. So you can see so many colors: orange, red, yellow. And then after that, it's important to introduce some concepts like benthos community. So to talk with students how rich diversity and high density of minerals, they are so important to make mineralization happens. And this is also very important for the carbon cycle. So it's important to connect these ideas to make the students understand how important is this benthos community in the sea. And after that, I developed some science activity if we have teachers listening to us now. So this science activity, it's a way to introduce some ideas about trophic levels and about the impact that we as humans are doing on the sea. So if we have like 10 chairs in a room, so 10 students, we can have each chair with a different name. So we can use the words that Francine told us. So we can have sponge, we can talk about plankton, we can talk about benthos, and suddenly we have 5 chairs or 10 chairs with different names. But then this activity, it's called music chairs. So we can put the music and we can have these words that we use in the class, but also you can see here on the other line, we need to introduce other words like plastic, pollution, water pollution, and global warming. So we are going to have these titles here, these words, and then we are going to have these music chairs. So each time we are going to put the music, and then the music is going to play. And after the music stop, we are going to take out, like here, for instance, the first one, Sponge. He's going to stop playing, and then we are going to introduce Plastic. And then we are going to have the second round. And when we came here in Turtle, we are going to stop the music, we are going to take out the turtle, and then we are going to introduce some pollution. And then on the third play, we are going to play the music, and we are going to take out plankton, and we are going to introduce water pollution. And suddenly, next to the end of this game, we are going to have many problems in different trophic levels. We are going to have so many plastic pollution, water pollution, global warming, and we need to discuss with students what is going on. So when we have water pollution, we connect the idea of water pollution with plankton or even with benthos. And this word is going to be out of the game. And in the end, we need to discuss with the students what is going to happen if we have a lot of pollution, what is going on if we have global warming in this area. And we can make this connection to understand the kind of pollution we are facing today with the problem we are having in plankton and benthos community. So thank you a lot for your attention. I'm going to share with you this class, the ideas, on the website of Polar Educators. And if you have any questions, please do not hesitate to text me. Thank you a lot for the invitation.
Thank you so much, Eileen. And as an elementary school educator, I can see using that in my classroom. That would be a really good application for students that could be adapted to different grade levels. So are there any questions for Eileen before we close out this evening? I know Julia Dooley, our head of Polar Educators International, she made the comment too, very good suggestions for educators to use in classrooms. So that's wonderful. And Gary also says, thanks for this idea. I think it can be utilized in many ways, Gary. I certainly agree.
Nice, thank you.
Okay. I'm going to show my screen here. And I want to tell you all, thank you very much for taking part in this masterclass series. Hope you all learned a lot tonight from Francine and Eileen about the science and then how to transfer that science into classrooms. And please stay tuned for more information about our next masterclass in early 2019. And thank you for the masterclass working group and the website working group, particularly Betsy, who's been invaluable in helping with all the technology and website application for this class. Thanks to everyone and have a good evening.